and neuroinflammatory cell traits. So just to give you a brief introduction of what microglia are. Microglia are the resident macrophages of the brain, and they perform a variety of functions, which includes migration and phagocytosis of debris, foreign bodies. And there is increasing genetic evidence, including at the level of common variants from US studies, but also from rare disease variants, that microglia really play a central role in neurodegenerative diseases, such as Alzheimer's disease. And we have seen that genes near these variants are often predominantly or exclusively expressed in microglia compared to other brain cell types. But we still have the unresolved problem of trying to link these genome-wide association study variants to target genes, and these to cellular phenotypes, and finally to see how this cause changes in disease risk. So first of all, because we're using iPS-derived microglia, I just want to convince you that the microglia that we are using recapitulates through microglia transcriptome and biology. So we wanted to see if we could improve differentiation protocols to get the best possible microglia. Some in our group systematically examined many cultural conditions. We evaluated cell morphology, viability, transcription of signatures. And he did this in the last part of the microglia differentiation process from the precursors. And after initially eliminating conditions that were not effective or that cause issues in the cells, we narrowed it down to 6 conditions and used single-cell RNA-seq data to do an automatic cell annotation of these cells. We wanted to check in which conditions cells have a more microglia-like signature and a reduced perivascular macrophage signature, which is a similar cell type that we're not interested in. We finally selected a protocol that yielded cells with a better microglia profile, while at the same time retaining a higher number of healthy cells. It has been published in scientific reports. Then from that, we took around 250 donors from the human-induced pluripotent stem cell initiative that have been differentiated in pools of between 15 and 72 donors to microglia. And from this, we subject this microglia to different treatments to just try to imitate the inflammatory challenges that they may suffer in the brain with interferon gamma and LPS. And we extract single-cell RNA-seq information as well as cellular phenotypes from 2 assays of microglia function. These are phagocytosis function and chemotaxis or migration. I'm going to focus on the phagocytosis assay in this talk. And from this, we wanted to see first if we could relate the variants that have been associated by US to neurodegenerative disease, to EQTLs, and see if we could find any sort of phenotypic associations as well from these assays, and relate this to these other 2 kinds of data sets. And then from this, we also have validated some candidate genes for Alzheimer's disease using CRISPR knockout screens. So first, from the single cell RNA-seq signatures, we investigated the presence again of the microglial subtypes and markers. We have been using the sets of markers from the recent publication by Mancuso, where he describes different levels of microglial subtypes. And we can see that for the untreated cells, we, in general, have a variety of signatures from the general microglia that we can see across the 3 different treatments, but from the untreated, we get an increased signature in general from disease-associated microglia across all the cells, except for 2 classes are more proliferative of like cycling cells. From the interferon gamma, we have an increase overall expression of HLA genes. And from the LPS, we have, again, some big signatures from proliferative cells. The rest of the cells resemble mostly cytokine response microglia, with some disease-associated microglia as well. And we have taken all these single-cell RNA-seq cells from the different treatments, and we wanted to see, in general, if we saw expected responses from the interferon and the LPS challenges. So we aggregated the UMI counts as pseudoballs per line, pools and treatments, and performed differential expression analysis on lines that present over 100 cells in the overall clusters. And we see that we get a huge number of differential expressions. From the gene set enrichment analysis, we can confirm the increased expression of interferon gamma and inflammatory response genes in both the interferon gamma and LPS treated samples. So here you can see, you say interferon versus untreated, LPS versus untreated, and interferon versus LPS. And we get that the interferon gamma-specific gene sets are more significant in the interferon gamma versus the untreated compared to the LPS samples. And in the LPS signature, we see also an increase in the NF-kappa B response, which has been linked to increased tauopathy on tau toxicity. And from this signature, interestingly, we also see increased candidate gene enrichment for neurodegenerative disease, particularly for Alzheimer's disease, in the LPS, but also in the interferon gamma signature as well. So one thing that we could do, and that we have done in this data set, is just to try to see if we could get an aggregated signature of risk for all the lines that we've got by calculating the polygenic risk scores for Alzheimer's disease. And the way that we did this was using PRECISE, which uses all the signatures from the GWAS summarized test from Alzheimer's disease. And after performing aggregation based on p-value and LD, we get a polygenic risk score that we then can standardize against the polygenic risk score from the 1,000 genome European subset. And you can see here how this distribution of the polygenic risk score compares to this 1,000 genome signature. We have a few donors that have higher, like more than 2 standard deviations away from the mean risk in this Alzheimer's disease signature, while the majority stay, in the average. And this polygenic risk score has been calculated separately for what we call the polygenic component without ApoE, and then we have incorporated the ApoE signature from the different alleles because of the massive effect that we know that these alleles have in increasing the Alzheimer's disease risk. So indeed, if you check the polygenic risk score of ranks of all the lines in order without ApoE, after incorporating this ApoE signature, you can see how they just move around a lot depending on whether they have particularly the very high combination of risk in this ApoE, the double E4 allele. That would be the one that confers the highest risk. And then from this, we wanted to see if we could find any sort of general transcriptome signature that could depend on this polygenic risk scores. So we did this again by aggregating the expression from pseudobulks by line and pools and the clusters, and then we tested this with including some of the covariates, including sex and pool as a random term. And we can find some differential express genes, even though the effects, as you can expect, are small. And we have done this separately for each treatment. And these effects are very robust, so we don't expect this to be due to technical and any sort of technical confounders, because we have redone this analysis and compare empirical p-values after shuffling the polygenic risk score values from the lines 100 times. And as you can see from these pink dots here, the significance of the genes are like mostly maintained after this shuffling. And then we wanted to see if we could see any sort of aggregate signatures from reactome hallmark pathways and all pathway analysis. And we see in general upregulation at the bottom polygenic risk score of the MYC targets, which are involved. MYC upregulation has been involved in increased proliferation. And we also see an upregulation at the bottom of the risk of the oxidative phosphorylation. And we find that quiescent microglia present increased oxidative phosphorylation metabolism. And in the literature, we can see some evidence that the microglia switch to the glycolytic metabolism, as opposed to this oxidative phosphorylation metabolism, when they are taken from samples from brains with Alzheimer's disease and Parkinson's disease, and in general, microglia that are present in these diseased brains. So this could indicate some role for these sets of genes from lines that have fewer overall risk of developing Alzheimer's disease, in having been part of a metabolism that is in general has been more associated with healthy brains. And finally, we have also seen from tests by LD score regression. We wanted to test with LD score regression whether we could see enrichment in the Alzheimer's disease heritability in any particular treatment. So the LPS or the interferon or the untreated lines compared to each other. And we have seen that for the LPS treated cells, we observe an enrichment in these lines that have in the genes, that are associated with high polygenic risk scores when compared to the other treatments. So we can see this in general for the LPS, and we see that this enrichment of the heritability is maintained when we condition the signal on the sets of genes from the untreated sample, but not when we see this conditioning on the interferon sample. So this is due, we think, because the signatures between the LPS and interferon are very similar, and we do see a high correlation in the T statistics of the genes between LPS and interferon in this analysis. But we find very interesting that in general, we tend to see that this enrichment of Alzheimer's disease heritability happens in the cells in response to neuroinflammatory challenges of LPS and interferon gamma. We then proceed to perform expression quantitative trait locus mapping, EQTL mapping with TensorQTL. And we have done this after selecting the optimal number of PCs that is different depending on the treatment. So we see that in general, we find an optimal number, like the highest number of EQTL-associated genes at very high levels of expression PCs fitted in the model. And these are some implications for the EQTL catalog mapping for other cell lines as well. We found, after talking to other people doing EQTL mapping in other cell types recently, that it's better to do it this way, because in general, people tend to fit between 5 and 15 PCs, but they may be losing some signals of EQTL associations if they don't go further than that. And indeed, we find between 4,000 and 5,000 genes, which is twice more than what was found in the analysis by the EQTL catalog. And these EQTL's effects are in general quite shared. So we find that the betas are in general in line, go in the same direction, and are within 0.5 in value between them, and they tend to be significant in the same way. And if we go even further in detail in this, we find in general EQTLs that are significant only in one of the treatments are around, the 300 to 500, and then we get some pairwise sharing between the different treatments. But we see more or less the same kind of effects between these. And then we wanted to see if we could colocalize these signatures with different sets of neuroinflammatory disease GWAS. So we have tested an Alzheimer's disease dataset with 83 associated loci. This is the latest one from Belenguez that we also use in the polygenic risk score analysis. And we have also tested ALS, MS, and Parkinson's disease. And except for ALS, they more or less have the same number of significant association in this GWAS. But despite this, the microglia colocalizations seem to be particularly enriched in Alzheimer's disease. We see a very high percentage of colocalizations. So we get 25% of all the loci from Alzheimer's disease was colocalized with the EQTLs in our data set for the untreated disease, and around between 15 and 20% for the other two treatments. And we see a smaller number of colocalization for the others, particularly for Parkinson's disease. And most of these colocalized genes tend to be somewhat shared. So we get a high amount of pairwise sharing between untreated and LPS, but also across all of them. But the untreated seems to have the highest number of specific signatures. Just to highlight a few of the interesting genes that show up in the colocalizations, we have found that both PI-LRA and the PI-LRB colocalize with Alzheimer's disease to different levels depending on the inflammatory trait. So it is, we find this to be interesting because we know from the literature that there are separate roles for these two genes in Alzheimer's disease. So PI-LRB expression increases in general inflammatory response, and the A gene has been shown to be to present protective variants that reduce the binding of the herpes virus and other viruses that have been associated with the increased risk of Alzheimer's disease. And they have also been associated with modified microglial phenotypes, including cytokine production and phagocytosis. But both of them are modulated by the same variant in interferon gamma response. And this seems to be, from what we gather from the analysis of the literature and open targets, this seems to be the first evidence of microglial colocalization in the genes, provides increased evidence from the locus to gene pipeline from the open targets target prioritization for the PI-LRB. And we also find a colocalization of TREM2 with a protective signal for Alzheimer's disease. So we find, again, very high posterior probability for colocalization for this gene. And if you're familiar with Alzheimer's disease, you'll be familiar with TREM2. It's an amyloid beta receptor. It's in the same microglia activation pathway as PI-LRB, linked through thyroid BP response, linked to alterations in, again, several microglial phenotypes. And we find that rare missense variants that are in high linkage disequilibrium with our EQTL associated variant cause, have a very high risk of frontotemporal dementia, Alzheimer's disease, and we see from the effect of this colocalized variant that this EQTL variant that increases TREM2 expression seems to be associated with reduced risk of Alzheimer's disease. And we will come back to this gene at the end of the presentation. So we wanted to, I wanted to present to you also how we perform the phagocytosis analysis. We're going to leave the migration analysis aside for a moment. And we get this phagocytosis analysis from the same microglia that have been differentiating the same pools, underwent the same treatments. And the way that this phagocytosis analysis works is we put human sushi cells labeled with M-cherry in the medium with the microglia. And then the microglia will eat these M-cherry labeled cells, and they will go through a FACS sorting. And then because of the change in the phagolysosome of the pH, these M-cherry labeled cells will change color. And then we can select them, select the microglial cells that we know have incorporated these cherry cells inside of them. We select them in this FACS bin, we sort them, and then we can also select only microglial cells that have not incorporated these labeled cells. So this way, with the change of the color inside the microglial cells, we ensure that we're not just sorting only the sushi cells, the labeled cells. So only microglia without phagocytosis or that have phagocytosis cells, and we can then perform whole genome sequencing on these bins. And the way that we test for this association between lines and phagocytosis is by performing donor abundance deconvolution by whole genome sequencing. And we can do this by leveraging the genotype information that we have from each donor and the minor allele frequency at each position of the variants that we know change between the different lines. And we can see whether we are observing a change in the abundance of certain lines in the low phagocytosis or the high phagocytosis bin. So we then can test for changes in donor abundance with a genome-wide association analysis. That didn't work very well because we didn't have the power, so I'm not going to go into that. But we have also done burden tests for these changes in donor abundance across the phagocytosis beams, and differential expression analysis across the phagocytosis beams, which is a bit of an indirect measure, because obviously the cells that we have the single cell RNA-seq data from, and these cells are not exactly the same cells, but it gives us a possible proxy. I will focus first on the burden test, but first of all, I wanted to highlight the main problem that we had with this is due to this issue that you probably have also seen. There's that a few lines take over the pools. So we see that even if we plate the lines in the pools in the same proportion, over time in the expansion, some donors will take over. And then during the differentiation and aging, this problem gets exacerbated. But interestingly, we found that at every step of the differentiation process, this direction of reduction or increase of the abundance of these cell lines can change direction. So, for example, we see that this line here through the expansion increases, but then in the differentiation it changes direction, and then it massively decreases compared to other cell lines that survive the previous expansion steps. And I'm going to show you first the genes that we saw were associated with these different proliferation steps, because I think it might be useful for you to compare in your own experiments. So we have measured this at different steps. We have measured the abundance at the iPS stage, at the macrophage precursor stage, and we have both young and old macrophage precursors, so they have been in culture for longer if they are old. And then also between, we have also measured obviously the abundance at the microglia step, so we can compare between the macrophage precursors and the microglia, and the macrophage precursors on the iPS. And the way that we have done this is we have annotated variants in each line from whole exome sequencing, and from the variant effect predictor tool, we have selected deleterious variants as those that had loss of function or misses pathogenic with a FRES score of 15. Then we have other missense variants that do not fulfill this, that are classified as missense non-deleterious, and also synonymous variants, just to compare how this significance of the burden test changes between deleterious and the other two categories, when we expect, obviously, for the synonymous not to show any sort of burden test association. This is the case, for example, for the comparison of iPS to macrophage precursor. So we have 3 significant associations from the burden test for the deleterious variants. And one of them, one of these genes is familiar B-core. So you can see that we have reduced proliferation. So that's what this scalar fraction means. Reduced proliferation with increased burden of these deleterious variants. So what we'll find is, if you have this deleterious variant in B-core, you'll have fewer proliferation at the stage of the macrophage precursors. And this is very interesting because we were able to recapitulate what we have seen in other proliferation analysis in other cell types. So we were very happy to see this. We also have one association by Barden test between the old and young macrophage precursors. So we see an increased abundance of lines in the old precursors that have deleterious variants in NEC11. So this the NEC11 protein is involved in the G2M checkpoint response to DNA damage and is upregulated in somatic cells with arrested cell cycle. So it is a consistent direction that if we see a higher burden of deleterious variant, we will see like probably a decrease in the expression of this protein, this gene, and higher proliferation. And then we get much more significant genes in the microglia versus macrophage precursor step. So we find 18 significant association. This is all after multiple testing correction, obviously. This also includes the gene that we chose to highlight because it codes for an adapter protein that has been previously associated with dopaminergic neuron differentiation. It went in that case in the opposite direction, but it is interesting that we sort of see, we could see the same genes having so different effects depending on the target cell line from the differentiation. And we also see other interesting genes like caspases that we know are involved in apoptosis. So that makes a lot of sense. And we can aggregate the signature of the previous associated genes to find a global score for increase or decrease proliferation. And that's what we have done here. In general, we see a slight global burden for all the deleterious variants, but the signature is much stronger when you aggregate the signals for the different like the different associated genes survive the multiple testing correction. This can be helpful to pre-rank candidate donors in future studies for inclusion or to try to pull them together to minimize the effects of donors overtaking the pools. And we have done also this burden test for the phagocytosis phenotype. Here you can see how these genes overlap between the different treatments, and we find one specific interferon gamma signature that was quite interesting. So this SPG11 gene is has been found to be involved in other neurodegenerative disease, so spastic paraplegia. So that's like a multi-system neurodegeneration disease. And a recent study found that a double knockout of the gene in microglia stimulated with interferon gamma presented increased phagocytic activity versus controls. So that would recapitulate this phenotype that we're seeing quite nicely. And then we also found the anti-inflammatory cytokine IL-37 to be significantly associated with the increased deleterious burden associated with increased phagocytosis, which goes in line with previous studies that link this gene to phagocytosis in monocytes and macrophages. And finally, we performed differential expression across the phagocytic range and compared the results with a CRISPR knockout coupled with the phagocytosis analysis on 251 candidate genes. Now, this CRISPR knockout analysis are very noisy, but we found from the top 5 genes that are ranked with the score here with the FDR correction, so like building a kind of like a T score from the beta and the p-value. If we compare it to the T-score from the differential gene expression across the phagocytic range, that 3 of these match the directionality, and these are the ones that so the ones that show like actual significance in the CRISPR knockout. And the most important one is TREM2, that follows the same direction, and we have seen consistently in different cell lines from the CRISPR knockout, different repeats with different cells, different experiments, that in our microglia, TREM2 knockout increased phagocytosis. So this goes against what we see in other CRISPR screen knockouts. But we have seen when investigating the literature that there is an inconsistency in the phagocytosis directionality with the expression of TREM2. So we have found from this review that aggregates the what we know about the TREM2 and phagocytosis directionality quite extensively, that in the early disease, TREM2 knockout tends to decrease phagocytosis, while in the late disease, TREM2 knockout tends to increase the phagocytosis. And we have seen also from the literature that the disease-associated microglia from the different microglia models resembles more late Alzheimer's disease. And you can see here also from the recent publication from Mancuso that with TREM2 knockout, we find that these there will be like a change between the disease-associated microglia to more homeostatic stage. We haven't tested this, but we're very interested in testing that. But what we know for sure is that our cells resemble much more disease-associated microglia by the differences of markers and by automatic annotation compared to the other cell types. So I think we have a very large body of evidence from our study, also supported by other studies, that we can see this complex behavior of TREM2 depending on how advanced the Alzheimer's disease stage is. this is all I have for you today. So I want to thank everyone in the group, Gosia, Andrew, which are my main supervisors, and everyone else that has helped me with this project and the funders. And I'll be very grateful for your questions. Thank you.
Thanks. Questions from the group now?
Maybe I can start by asking a very general question. So when you initially start doing the analysis of microglia using gene expression, microglial states, you at the end you cited that paper Mancuso et al. and from de Stroeper's lab, and also there's a lot of work from the Broad on Alzheimer's microglial states. And I was wondering if there's a kind of consensus between the different groups as to how many microglial states can be powerfully analyzed and shared between labs at the moment. I mean, is that a good question or not really?
I think it really is. I think it's very relevant because at this moment and since last summer, there has been a huge effort led by the Strooper and other people to try to get a consensus of microglial subtypes, because it does seem like every lab that works with microglia has their own favorite classification. I try not to do that with my cells because I don't want to add yet another microglial subtype to the pool of dozens of subtypes. So I try to focus on their classification, because from the paper of the from our previous paper from our group, where we describe the differentiation, how we chose the differentiation media, we found that Mancuso's cells were the most similar to ours. So we wanted to keep using that classification, or at least a classification that made sense, considering our own comparisons. Does that answer your question?
Yeah, I guess so. I mean, I think it's happening sort of all in different fields. You know, the RNA transcriptomics lends itself to this kind of analysis. So then you get into an interesting place where you can ask, is there co-regulation between the microglial classes that you have and different cell types in the brain itself? And you imagine that they're going to be communicating with each other, so there should be cross correlation, and in principle it should be extremely powerful system to ask about mechanism. Maybe it's just at a level we haven't quite grasped yet. I'll just leave it at that way.
I know that people like at Sanger are setting up now experiments, have been doing so for a while, with trying to understand this cross talk that you referred to, doing co-cultures between the different brain cell types to try to understand that process. So yeah, I think you're absolutely right that is something that we should be investigating, and that people, not we, like I am not, but people are investigating right now.
It looked like a lot of the associations about 3/4 of the way through your talk were based on the number of estimated deleterious effects at particular loci. Did you do a similar analysis across common variants in the pool, or did I miss that?
So when I presented the sort of overall schematic of the phagocytosis analysis, I hinted a bit at that, but I did not want to get into that in this talk because the reality is that we were severely underpowered due to the number of lines and the dropouts of the lines, how some overtook the pools.
Right.
That means that in the end you end up having, like, we started with 261 and ended up with 140 best case scenario, and that made things really, really noisy. So we didn't get any significant genome-wide association. Okay. So we are looking into that because we know, even if we don't have anything that is genome-wide significant, the shapes of the Manhattan plots look good, look reasonable. So there may be some under significant signal that is true in there.
Yeah.
But in 45 minutes, I think that would have been like too much.
Gotcha. Yeah, so the dropout of cell lines really killed you there, huh?
Yes. Well, I think that in general, when we started doing this analysis, we didn't know how strong the effects would be. We know that in general for the common variants, we expect the effects not to be obviously strong. So I think that the burden tests are more useful in this experimental setup. Yeah.
So a lot of the other speakers in this series that have been growing lots of cell lines together have experimented with the stage at which they mix the cells.
Yeah.
And given the very detailed plots of when you're losing and gaining lines, it looks like you mix them extremely early. Is that correct?
At the IPS stage.
Right.
Yeah.
Have you ever tried just to mix them after? I guess it's a lot more work to grow 260 lines separately is the point there, right?
Yeah,
there'll be 260 differentiations.
I wish the wet lab scientists would be here with me, but yeah, I can't answer that. I think logically it would be a huge amount of work for them, therefore very difficult logistically to support
you said
that.
there were 16 pools, right? So it'd be 16 differentiations versus 260. So
yeah,
that's exactly our problem too, and that's the answer. That you cannot. I mean, as soon as you scale up, it's unmanageable.
Right.
But wasn't that a remarkable part of your story, really? That you had assigned different risk profiles to the behavior of the cells at different stages in this protocol. Did I get that right? Was I following you? I thought it was really quite astonishing that what you were saying, really. That you had that profile of different individuals, let's call them, expanding at different times in the protocol. And then you followed that with like 5 minutes of discussion about which genes were responsible at these different stages. Isn't that right? Wasn't that what you were telling us?
Yeah.
That was quite spectacular.
Yeah, well, the thing is that part of, it's a shame because I also think that it's very interesting for the community and for the publication it will obviously be part of it because I think it's super useful for everyone. But that it's a shame because that wasn't really the point of the experiment. It's just a small subpart of the analysis. But yeah, I
think you're being modest.
It did work quite well.
I think it's really cool. And if we can generalize for a minute, then these approaches, it seemed like a lot of the alleles that you had found that were conferring this phenotype, these different phenotypes, were known. known to be involved in different, let's say, different aspects of cell replication.
Yeah.
So it seems then that you could almost, I'm thinking of an Alon paper about 2 years ago where he showed that actually cell cycle genes, kind of canonical cell cycle genes, are not really involved in cell fate. And I guess maybe that was in the frog. And but it seems like an area that really is worth a lot more exploration. How many different kinds of cell cycle are there actually? And in your system, it seemed like approachable. It's fun to talk. Thank you for your presentation.
Thank you.
Just I wanted to mention something very brief. I was very happy to see BCore again on as an highlights, because in our paper we saw that somatic mutations in BCore fails to produce dopaminergic neurons, and I see that in a completely different lineage it also has an effect. In this case, I think it's like at progenitor population, right? You said so.
So it's in the macrophage precursors, yeah.
Or in the precursor, yeah. So I think in that case it's the opposite, because in our progenitors it was affecting the proliferation rate. I think it was increasing it. And in this case, it's like something opposite, no?
Do you mean opposite direction or like a different cell type? Or a different part of the differentiation process?
Yeah, it's maybe it's not comparable, but I mean that it's curious that the same mutations in the same lines might cause different phenotypes in completely different cell types. Might be still be relevant. That's, I mean,
it goes in the same direction from what I could check from your paper, but it is a different cell type. So
but it's the same direction.
Oh, cool.
Same direction.
Yeah.
Okay. Thank you so much.
Thank you.
No other questions, we can wrap up. Really appreciate it. Very cool work. And
Thank you.
like you
Thank
guys
you
are
very
all
much.
highlighting, when we get back to the practicalities of growing these cells, we have to somehow overcome the practical issues of growing many, many cells together in a way that doesn't lose half of them along the way, right?
Yeah. It's really a trade-off with the batch effects and everything. So yeah.
Thanks again. See you next month, guys.
Thank you so much. Bye.
Bye.
Thank you.